Greetings, my name is CEDENO-BAEZ, and during this video presentation, I'll be talking about my summer research performed at the University of Iowa as part of the 2023 Summer Research Opportunities Program. During my research experience, I was working under the mentorship of Dr. Ethan Anderson in the Department of Pharmaceutical Sciences and Experimental Therapeutics at the College of Pharmacy. I also had the great pleasure of working along with Rachel Crawford, a third-year PhD student there. The main goal of our project was to develop an assay to characterize monoamine oxidase A and B activity in biological material by using various catecholamines such as norepinephrine, dopamine, and tyramine, which acts as a catecholamine releasing agent. After many attempts to figure out how to achieve our goals for the project, we were able to succeed in the end, and this allowed me to present it at a poster presentation conference held at the University of Iowa. So let's begin talking about the project, which is about monoamine oxidase activity in cardiac tissue from mice with cardiomyocyte MAO-A deficiency. To start off, I'd like to define what monoamine oxidases are. So MAOs are enzymes that are found on the outer membrane of mitochondria. They exist in two different isoforms. We have the MAO-A isoform and we have the MAO-B. These MAOs play a critical role in the heart by metabolizing the neurotransmitter known as norepinephrine, which is an important chemical messenger that regulates heart rate and contractility. While this metabolism is important to manage norepinephrine signaling, it actually generates damaging reactive oxygen species, including hydrogen peroxide and dopagal, which is a highly reactive catecholалdehyde. And this is actually the image you can see on the bottom. You can see a better explanation of how when norepinephrine gets metabolized by the MAOs, it will cause the creation of hydrogen peroxide and dopagal, which can bring damaging consequences to surrounding proteins. We think this is especially important in the heart because norepinephrine, not only is it a signaling molecule for the heart, but it is also a substrate for MAO-A, hence suggesting that MAO-A inactivity can protect the heart from catecholamine-induced stress. In previous studies, both MAO isoforms A and B have been linked with cardiac diseases, and our group was able to report that mice with cardiomyocyte-specific MAO-A deficiency appear to be protected from suffering ventricular arrhythmias. So with this arises the question of determining if targeting MAO-A with drugs could be useful in treating heart disease or any other cardiac disorder. That is why for our project, we decided to use a mouse model that does not have MAO-A specifically in its cardiomyocytes. That way we can see if reduced activity in the heart is actually protective against cardiovascular disease. And we did this by creating an experimental assay in which we analyzed the activity of both MAO-A and B isoforms in heart tissue obtained from wild-type mice and MAO-A deficient mice, which we'll refer to as our knockout mice. So to run our assay, we used a total of 17 samples from both wild-type and knockout mice, from which we obtained pieces of heart tissue that were used to prepare our cell lysate using our TEE plus tween buffer. We then homogenized each of these samples, and these were then centrifuged for a total of 10 minutes at a temperature of 4 degrees Celsius. After centrifugation, we would let the homogenate sit on ice for a period of 15 minutes before taking out the supernatant. And once we obtain our cell lysate, we would prepare a 1 to 10 dilution using our PBS buffer. So we would add 100 microliters of the homogenate and 900 microliters of the PBS to prepare our dilution. This dilution was then separated into 4 different tubes to allow our inhibitors to incubate properly. So these tubes, one contained no inhibitors, one had chlorigylene, which is an MAO-A specific inhibitor, another one had selegiline, which is an MAO-B specific inhibitor, and lastly, we had a tube that contained both chlorigylene and selegiline. It's important to note that both of our inhibitors were being used at a concentration of 20 micromolar. So once we had all that ready, we prepared a master mix that contained the PBS buffer plus Amplex Red, which is a reagent developed specifically to detect hydrogen peroxide. And we also added horseradish peroxidase and superoxide dismutase. With all of this ready, we used a 96 well plate to get our assay ready, to which we added our lysate substrate, which was tyramine at a 1 micromolar concentration, and we also added our master mix. So once this was all ready with our lysate samples, we then proceeded to measure its fluorescence for 30 minutes. So here we have the hydrogen peroxide standard cube we used to analyze our data. Basically, the way we obtained our standard curve was by preparing a plate similar to our assay, where the PBS buffer was replacing the lysate. And we also added various concentrations of hydrogen peroxide directly to the wells, and we measured that. The next image explains how we obtained the fluorescence measurements we needed. So, because one of the products created as part of the MAO metabolism is hydrogen peroxide, we use this in our favor to obtain the fluorescence we need by making that hydrogen peroxide react with other reagents. So we made this possible by using Amplex Red, and we also used horseradish peroxidase so that it could react with the hydrogen peroxide and form the fluorescent molecule known as resorufin. So the more hydrogen peroxide that is being made, the more fluorescence we'll get from this reaction. So onto the results. Here we have different graphs demonstrating the MAO-A and MAO-B specific rates. And we decided to divide these according to the mice gender because the gene MAO is X specific. It is found on the X chromosome. And as you may recall, males have one X chromosome while females have two. So based on that difference and sex differences in general, there is a high chance of there being a difference when looking at these rates. Now, it doesn't really show a huge difference on our data, considering that we used a small amount of samples, but the possibility is still there. So, as you can see on the graphs, while the difference between wild type and knockout is not significant because the p-values obtained did not meet the standard, you can still see that there is a trend of reduced activity in the knockout samples. Because typically for there to be a statistically significant difference between two groups of data, the p-value, which is the number above the line on our charts, it needs to be less than 0.05. And since all of our p-values are above that number, they are not statistically insignificant. But regardless of this, you can still see the trend in the reduced activity. So here we have the rates of the males, for which we obtained a p-value of 0.16 for the MAO-A rate and a p-value of 0.17 for the MAO-B rate. Next, we have the females. Again, we can see the same rate of there being a reduced activity on our MAO-A deficient mice. So on our MAO-A rate, we have a p-value of 0.24 for the first one and 0.16 for the MAO-B rate. And lastly, we have charts containing the rate of hydrogen peroxide production on all of our samples, regardless of the mice gender. And here we can see that we obtained a p-value of 0.13 for the MAO-A rates and 0.09 for the MAO-B. And if you can notice, here it is much more noticeable that there is a reduced activity on our knockout mice. This graph of hydrogen peroxide concentration in picomoles versus time in seconds is what shows our major findings during this assay. So we can clearly see a reduced MAO activity when inhibitors are present. To explain this a little better, we can see that the samples containing no inhibitors had a pretty good rise in terms of hydrogen peroxide concentration, and we can actually see the same result with the chloridolene, considering that the mouse model we used was MAO-A deficient, and the isoform that chloridolene targets is MAO-A. So with nothing to target, our hydrogen peroxide concentration will still rise. However, that is not the case with the soligelene and the one containing both inhibitors, which means that MAO-B was successfully inhibited by the soligelene. And as a result, what this demonstrates is that the change in fluorescence being measured is actually MAO specific. So for future directions, we weren't able to get the assays to work with catecholamines such as norepinephrine and dopamine because of all the background fluorescence these created by reacting with the amplex red and horseradish peroxidase themselves. So taking this into consideration, a different assay could be performed in the future by using other MAO-A and MAO-B specific substrates, and that way test their specific activity as well. And to conclude, I am proud to say that we successfully developed an experimental assay to confirm the kinetics of both MAO isoforms in cardiac tissue from our MAO-A knockout mice by using tyramine as our substrate. For acknowledgements, funding was supported by the University of Iowa's Summer Undergraduate Research Program and the American Heart Association's Strategically Focused Research Network. We have the award number right there, and I would like to give special thanks to all of the members of the Anderson Lab for offering their guidance during my time working on the project. And I would also like to thank the Summer Research Opportunities Program for allowing me to have this amazing experience. Here are the references I used for the project. I truly hope you enjoyed this presentation, and if there's any doubts or questions regarding the project, please feel free to contact me.